Chapter 7, Mutations of Genes. The learning objectives for this topic are to be able to define mutation, define point mutation, and describe 3 types, list 3 effects of mutations, discuss how different types of radiation cause mutations in a genome, describe 3 types of chemical mutagens and their effects, discuss the relative frequency of deleterious and useful mutations, describe light and dark repair of pyrimidine dimers, base excision repair, mismatch repair, and the SOS response, contrast the positive and negative selection techniques of isolating mutants, and describe the Ames test and discuss its use in discovering carcinogens. The overview for this topic is types of mutations and their effects, mutagens, frequency of mutations, DNA repair, and identifying mutants, mutagens, and carcinogens. First, let's talk about what a mutation is and their effects at the same time, because they naturally go together. In the normal course of things, DNA will be replicated so that it is an exact copy of the parental DNA. Then we get an mRNA transcribed from the template strand, and that mRNA is translated into a polypeptide, of which we're using the labels normal, or we also use the term in biology wild type, or what's seen most frequently in the wild. Now, if we change one nucleotide base, we also call these base pairs. We call this a point mutation or a substitution mutation. Now, the first point mutation that we're going to talk about is a silent mutation. There has been a change in the DNA strand. Any change in a DNA strand is a mutation. If we get a mistake or a change in the mRNA, that's a temporary copy. It's not a mutation. So here we've had an A change to a G, which changes the base pair, but it doesn't change the amino acid sequence. So we call this a silent mutation. There has been a change in the DNA sequence, but there is no change in the amino acid sequence. Now, what if there is a change? Here we've changed the G to a C so that it reads CTG instead of GTG, and instead of an alanine we get a glycine. So we get a slightly different amino acid sequence. This is a missense mutation. We've had a change in the amino acid sequence. Sometimes we can get a change that leads to an early stop codon, and we call this a nonsense mutation. The polypeptide synthesis stops early. That basically kills the gene, is the way that I put it. The gene is still there, but it is not producing a usable protein. Now, what if instead of point mutations or substitution mutations, we're either adding or deleting? Okay, here we're adding a nucleotide, here we're deleting a nucleotide. This causes frame shift mutations. Everything down the line changes. The reading frame changes, so that everything from here on is different. Now, this can cause a catastrophic change in the protein. Very, very rarely is this helpful. Sometimes a missense mutation can be helpful, not very often, but this is even more rarely helpful. Usually it causes a problem for the organism. Same with a deletion mutation. If you remove one, it once again shifts the reading frame, and everything downstream is different. These mutations can be caused by any number of things, and we call them mutagens. So radiation is a famous one. It comes in 2 types: ionizing radiation, or radiation that causes ions. These ions go around disrupting DNA, and in worst case scenario it can break the sugar phosphate backbone and completely break up the chromosome. That's bad. That's why ionizing radiation can kill. Ionizing radiation is gamma rays, beta radiation, X-rays, that kind of thing. The other type of radiation is non-ionizing radiation, or radiation that doesn't produce ions. So UV radiation is an example of non-ionizing radiation. Remember that it's light that is above or has shorter wavelengths than violet. And what happens is it comes in and it causes, particularly in the case of when you have two thymines that are next to each other on a strand of DNA, it'll break the hydrogen bond between the T and the A and cause the thymine to form a bond with the thymine next to it. We call this a thymine dimer. And it causes a bulge in the sugar phosphate backbone. Well, DNA polymerase can't get its active site over this bulge. So it will either halt and then skip over the bulge, causing a deletion mutation, or it'll stick here and kind of stutter and then hop over, causing an addition followed by a deletion mutation, both of which cause frame shift mutations, which are really bad. Having a thymine dimer form during DNA replication is really bad. Let's move on to other mutagens, namely chemical mutagens. There are three types: nucleotide analogs, nucleotide altering chemicals, and frame shift mutagens. Let's start with the nucleotide analogs. 5-bromouracil is an analog to thymine. You can see that it looks exactly the same, it just has a bromine here instead of this methyl group. What it looks like to the cell, though, instead of looking like a thymine, it looks like a cytosine. So we have a change from a TA pairing to a CG pairing, and this causes a substitution mutation. Now, I want you to keep nucleotide analogs in mind for when we talk about antimicrobials, specifically chemotherapeutic agents that we would use inside of the body. Next, nucleotide altering chemicals. Nitrous acid and aflatoxins are both chemicals that will come in and alter the base of a nucleotide and cause it to switch from one base to another base. So it does approximately the same thing that nucleotide analogs do, but they are chemicals that are doing it to DNA that is already in place in the cell, whereas nucleotide analogs cause mutations in replicating cells, as the nucleotide analog is inserted in the place of a nucleotide. Next up, frame shift mutagens. The example that I'm using is benzoprine, which is found in cigarette smoke. Also, when we're in lab, make sure you're careful with the DNA stains that we use in gels, because they are also frame shift mutagens. What happens is they get inserted into the double strand and they cause a bulge, kind of like the thymidine dimers. And the same thing happens. DNA polymerase can't get its active site over the bulge, so it either causes a deletion mutation or a mutation followed by a deletion mutation. In either case, it's not good. Now that I've gotten you all scared about mutagens, let's talk about frequency of mutations. There is a baseline rate of mutation in any population of organisms. 1 out of every 10 million genes contains an error. And this comes from mistakes that are made during DNA replication. They can also happen from naturally occurring mutagens. But mutagens typically increase the mutation rate by 10 to 1,000 times, or 1 out of every 10 to the 6th or 10 to the 4th genes. This isn't going to increase the mutation rate. Regardless of whether it's the background mutation or an increased mutation rate, you do have changes in the organism. Now, in bacteria, that can cause either death, or it can lead to a bacterium that doesn't have an enzyme anymore that it needs to make, say, an amino acid, so it has to import that amino acid. It may lose a necessary pathway. That's how we got heterotrophs, when originally all organisms had to be autotrophic, and so on and so forth. And occasionally you will get a benefit, something that makes the bacterium bigger, better, stronger. In humans, when you're exposed to mutagens, because we're multicellular organisms, it can cause disease such as cancer, which is not seen in bacteria and single-celled organisms. Cancer only exists in multicellular organisms. That's where one cell in the community decides not to play nice and causes problems for everybody else. That's why we try to limit our exposure to mutagens, be they ionizing radiation, non-ionizing radiation, or chemical mutagens. The FDA is very involved in limiting the number and amount of chemical mutagens in your foods. Also, another thing to keep in mind is, especially when we talk about a topic a bit later, researchers use mutagens to knock out genes so that we can study pathways, so we can study the interactions between genes that we don't necessarily know what they do, and phenotype, which we can take a look at. So, all of that is important in mutagens increasing the frequency of mutations. Now, it doesn't matter whether we're talking about a multicellular organism like a human or a bacterium, you're going to be exposed to mutagens. You're going to have mistakes in replication. How come we don't all die? How come we're not all mutating at a fast rate? And how come we don't all have cancer if you're multicellular? Well, the answer is cells are set up to repair their own DNA. This damage happens. Those organisms that did not have good DNA repair mechanisms died. So let's take a look at the main DNA repair mechanisms. First, let's look at light repair. Okay, we've had UV come in and create a thymidine dimer. When visible light comes in, specifically blue light is right next to UV, it stimulates light repair. And the light repair activated enzyme comes in and removes the thymidine dimer by snipping this improper bonding between the two thymines, and so that they pair bond across the strand with the adenines. Very nice, very simple. Now there's also dark repair, because if the sun's not shining and the cell realizes it has a thymidine dimer, then it still has to fix it. So dark repair, DNA enzymes come in and remove the entire lump along with neighboring nucleotides. Then DNA polymerase 1, you remember DNA polymerase 1, it's the one that replaces the RNA primers and replaces them with DNA. Does approximately the same thing here. Comes in and fills in the gap. Then ligase comes in and fills in the nicks. So you can have repair whether the sun is shining or whether it's at night. There's also base excision repair. Okay, suppose DNA polymerase made a mistake and put in a G when it should have put in a T. Well, these enzymes, base excision repair enzymes, can come in, remove the oops, and it can tell which strand is older. So it takes a look at which one is the new strand, takes out the mistake, and replaces it. Now you may ask, how does the cell tell which one's the old one? The original strands get methylated or get other markers that allow the cell to tell which one is the parental strand, because you don't want to be replacing the wrong nucleotide. That would lead to a point mutation. And lastly, mismatch repair. So we've got the same situation here. We've got a T trying to bond with a G. It's not working out. Mismatch repair enzymes, instead of taking out just the mistaken one, takes out a whole chunk, and then DNA polymerase 3 comes in and repairs the gap. Ligase comes in, fills in the nick. As I alluded to before, there are times when we want to identify mutagens, and since mutagens are often carcinogens. So let's talk about identifying mutants. Suppose I'm studying penicillin resistance in bacteria, in which case I would want to be able to come up with more cells that are penicillin resistant so that I can do my studies more easily. So here I've got a population of bacterial cells, one of which in this entire tube is penicillin resistant. So I would plate these onto agar. I would have a medium without penicillin. so both types of cells are going to grow, and then I have a medium with penicillin, and only the one cell that is resistant to penicillin is going to grow and divide and form a colony. Well, I could take that one colony, put it into broth, grow it, use it. But I want to increase my population, look at different ways the bacteria can become resistant to penicillin. So I would put a mutagen in to increase the mutations, then I would get more cells that have penicillin. Now you can do the exact opposite. If I'm studying the pathway that produces tryptophan in bacteria, I could put a mutagen into the colony to knock out genes or the gene that is involved in producing tryptophan, which, by the way, it's going to be more than one gene, and then I can put it in a medium without tryptophan and with tryptophan and look for who's missing. Now this diagram, where we were looking for the organisms that are penicillin resistant, this is a direct method of identification. My example with tryptophan is an example of indirect identification, where I'm looking for who's missing. Now, on to the Ames test, which is currently being used to screen all food additives, anything that's going to be used for human use, close contact, to see if it's a mutagen, with the aim toward, if it is a mutagen, a strong mutagen, then we're going to take a look at it and see if it's carcinogenic in multicellular organisms. Remember, bacteria can't get cancer. They're single-celled. So what we do in the Ames test is we take our test organism, which is a strain of Salmonella that has a mutation so that it can't produce its own histone, which is an essential amino acid for making proteins. So we take the experimental tube and we put the suspected mutagen in. We'll say it's red dye number 5. And then we would put liver extract in. Well, the question is, what are we doing putting liver extract in? Chemicals get changed by your liver. So if we're testing a drug or a suspected mutagen, we want to see what's happening to the suspected mutagen once it's been through the liver. So we're adding liver extract to put in liver enzymes that are going to do that. In our control tube, we put in our His-negative Salmonella with the liver extract. So we've got everything the same except for the suspected mutagen. We shake it up good. We put it on medium lacking histidine. For our control, occasionally we're going to have what are called reversions back to Hist-positive Salmonella. In other words, they're going to be able to grow, but we're not going to get very many. If the suspected mutagen speeds up the process so that we have more colonies of Hist-positive Salmonella, it's no longer a suspected mutagen, it's an actual mutagen, and it goes on for further testing to see if it's a carcinogen in multicellular animals. That's it for this topic. Here's a table showing the different types of point mutations and their effects, and here are reminders of the learning objectives for this topic. Remember to write down any questions that you have.